మన చాప్టర్ ఐసిఎస్సిలోని ఫస్ట్ చాప్టర్ బయాలజీలో సెల్ ద యూనిట్ ఆఫ్ లైఫ్. సెల్ ద స్ట్రక్చరల్ అండ్ ఫంక్షనల్ యూనిట్ ఆఫ్ లైఫ్. సో యూజువల్గా ఏంటంటే మనకి సెల్ అనేది వెరీ ఇంపార్టెంట్ కాంపోనెంట్. సెల్ని ఫస్ట్ డిస్కవర్ చేసింది యాంటన్ వాన్ లివెన్హుక్. లివింగ్ సెల్ అసలు ఫస్ట్ సెల్ అనేది మైక్రోస్కోప్ కింద చూసింది రాబర్ట్ హుక్ ఫస్ట్ చూసింది డెడ్ సెల్, కాక్ సెల్స్ని మైక్రోస్కోప్ కింద చూసి, అది కాక్ సెల్ అంటే ఓన్లీ మనకి సెల్ వాల్ ఉంటుంది, మనకి ఎటువంటి ప్రోటోప్లాస్ట్ కానీ ప్రోటోప్లాజం కానీ సైటోప్లాజం కానీ ఏమీ ఉండదు. సో కాబట్టి ఆ ఎంటీ స్పేసెస్ని తన మైక్రోగ్రాఫియా బుక్లో ఫస్ట్ సెల్ అనే వర్డ్ ఇచ్చాడనమాట. సో యూజువల్గా సెల్ అనేది ఎంత ఇంపార్టెంట్ అంటే, సో సెల్ అనేది ఇట్ కెన్ ఏబుల్ టు లివ్ ఇండిపెండెంట్లీ. సో దట్స్ వై కింగ్డమ్ మొనెరా యూనిసెల్యులర్ ఆర్గానిజమ్స్, కింగ్డమ్ ప్రొటెస్టా యూనిసెల్యులర్ ఆర్గానిజమ్స్, యూనిసెల్యులర్ ప్రోకారియోటిక్, యూనిసెల్యులర్ యూకారియోటిక్, బోత్ ఆర్ కమ్స్ అండర్ సపరేట్ కింగ్డమ్స్. సో సెల్స్కి సపరేట్ ఎబిలిటీ ఉంటుందన్నమాట రీప్రొడ్యూస్ చేయడానికి, ఎబిలిటీ టు సర్వైవ్ ఇన్ హార్ష్ ఎన్విరాన్మెంట్, వాటికి అవి రెప్లికేట్ చేసుకోవడానికి, ఫుడ్ని గ్యాదర్ చేసుకోవడానికి, రెస్పైర్ చేయడానికి, ఫోటోసింథసిస్ చేయడానికి, సో ఇండివిజువల్గా కూడా చేయగలవు. సో కాబట్టి సెల్ ఈజ్ అ బేసిక్ స్ట్రక్చరల్ అండ్ ఫండమెంటల్ యూనిట్ ఆఫ్ లైఫ్. ద స్ట్రక్చరల్ ఫంక్షనల్ యూనిట్ ఆఫ్ లైఫ్ ఈజ్ నన్ అదర్ దాన్ ద సెల్. సో వితౌట్ సెల్ ఆర్గనైజేషన్ లేకుండా మనకి లైఫ్ లేదు. అందుకే వైరస్ని లివింగ్ థింగ్ అని చెప్పము, నాన్ లివింగ్ థింగ్ అని చెప్పము. నెయిదర్ లివింగ్ నాట్ నాన్ లివింగ్ అది. ఫర్ ఎగ్జాంపుల్, ఆల్ ఆర్గానిజమ్స్ ఇంక్లూడింగ్ అవర్ సెల్స్ ఆర్ మేడప్ ఆఫ్ మైక్రోస్కోపిక్ సెల్స్, right, which are made up of small cells. These cells perform activities which contribute to the overall task of an individual. It also to note that every organism starts its life as a single cell. So ఒక సింగిల్ సెల్ నుంచే వాటి లైఫ్ని స్టార్ట్ చేస్తుంది. ద సేమ్ సెల్ అండర్గోస్ రిపీటెడ్ డివిజన్ అండ్ గివ్స్ రైస్ టు లార్జ్ నెంబర్ ఆఫ్ మాస్. సో ఇంక్రీజ్ ఇన్ మాస్ అండ్ ఇంక్రీజ్ ఇన్ నెంబర్ ఆఫ్ సెల్స్ ఆర్ ద ట్విన్ క్యారెక్టర్స్ ఆఫ్ సెల్. So by increasing number of cells, the mass will be increased. So by increasing number also will be increased. The same cell undergoes repeated division to give large mass of cells. Further, the cells get specialized various tasks. Those are support. So cells are used for supporting. Certain cells are used for producing digestive enzymes such as trypsin, trypsin, chymotrypsin, carboxypeptidases, pepsin. So all these things are and give support producing digestive enzymes and carrying of photosynthesis. So mesophyll cells are the chief cells for performing photosynthesis. So cells, a basic study in biology. You have already read about cells in some detail in class 9. So ఇప్పుడు class 10లో ఏంటంటే, it is necessary to refresh our knowledge for a proper understanding of several aspects of biology which specially constitute the class 10 syllabus. So main points about cell is all living organisms are made up of cells. Without cells there is no life at all. So cellular organization is the true characterization of living. The cell is the structural and functional unit of the body. So the structure and function of the body is made up of cells. All living organisms develop from pre-existing cells. Okay, all living beings develop from pre-existing cells. So this was contributed from Rudolf Virchow, which is omnis cellula e cellula. All the existing cells came from pre-existing cells. Cells how numerous? So larger an organisms, greater the number of cells. Single cell. Many small organisms are made up of just one cell, one single cell only. For example, bacteria are single cell, amoeba is single cell, paramecium is single cell, yeast is single cell, euglena is single cell, dinoflagellates are single cell, diatoms are single cell. Some very small organisms are made up of few hundreds or few thousands of cells. For example, Spirogyra, which is filamentous, Volvox, this is also this is colonial. So these are small organisms, but they have few hundred and thousands of cells, which makes a colonial or filamentous form. And next, multicellular. Most the plants and animals we see around are made up of billions and millions of cells. So for example, humans, mango, which is made up of millions and billions of cells. Cells, how small it is. Cells are very small and can be seen only with a microscope. Without microscope, we cannot see a cell. We can see with compound microscope. Smallest cells are certain bacteria, RBC. You know, the bacteria would be the size of 0.3 to 0.5 micrometers. Okay, and the RBC will be 7 to 8 micrometers. And the cells in the human body, the longest cell is the nerve cell. The largest cell is the bird's egg. Actually, central large sphere will be there, ostrich egg. Before development begins in it, it is the largest single cell of the living world today. So by today, the ostrich egg is the largest cell ever. The white albumin of the egg and eggshell are the extra parts added to the actual egg. Okay, as it passes down the reproductive tract. So an average sized adult constitutes approximately 1000 million cells in the whole body. 10,000 million nerve cells in the brain cortex. 25 million RBC. 30,000 million WBC. Coming to the cell shapes. Different shapes of cell are often related to the different functions they perform. For example, human RBC. It is circular, biconcave. For example, if you take frog, in the frog it is circular. In the human it is biconcave because there is no nucleus. So for easy passage through the blood capillaries as to transport, that's how it will attain the circular and biconcave shapes. White blood cells. White blood cells are amoeboid. So amoeba-like movement with pseudopodia. So that can squeeze out through the capillaries. And nerve cells has the long, it can conduct, nerve cells has a property of excitability, elasticity, and it has the excitability, elasticity and irritability from different parts of the body, from brain and vice versa. God cells are the somatal pore in the leaves, are bean shaped. It will open and close. So that's how the transpiration will happen through the stomatal pore, and carbon dioxide and oxygen will happen through the stomatal pore. Remember, plants don't have any sort of specialized organs. Organ systems are not there for transporting or for coordinating all those functions. Those have only organs. So stomatal complex is stomatal apparatus is an organ, not an organ system. So what you see here in the left side is the animal cell. The right side is the plant cell. You can easily identify with certain cell organelles. For example, if you take mitochondria. Mitochondria is present in both as well as plant as well as animal cell, because respiration is the common thing that has to be takes place in both plant as well as animal cell. And Golgi body. Golgi body will be both in plant cell as animal cell, but in animal cell we call Golgi body. In plant cell we call it as dictyosomes. And centrioles. Centrioles are present in animal cell. It is absent in plant cell. In plant cell there is no centrioles or centrosomes. And we have the smooth endoplasmic reticulum as well as rough endoplasmic reticulum. Same smooth and rough endoplasmic reticulum is present in the plant cell as well. In animal cell also we have both. Lysosomes. Lysosomes are present in the plant cell as well as animal cell. So which actually contains, but more prone in the animal cell than in plant cell. And we have the cell membrane is present for the both plant as well as animal cell. And we have the nucleus. Nuclear pore complex, which is 10 to 50 nanometers in diameter, and we have this nuclear pore, nucleolus, nuclear membrane, mitochondria, cytoplasm, right? And we have the cell wall, cell membrane, rough endoplasmic reticulum, vacuole, large central vacuole, mitochondrion, and the cytoplasm as well. Right? So remember, the main difference between plant cell and animal cell is lacking cell wall. And coming to the plant cell have the cell wall, and animal cell have centrioles and centrosomes. Plant cell are devoid of centrioles and centrosomes. Animal cell is lacking large central vacuole, but plant cell have large central vacuole, pushing the nucleus towards one side. So these are the changes. The gross structure of the cell. First thing is cell membrane, or we call it as plasma membrane. This cell membrane or plasma membrane encircles the cell. It is the living membrane and having fine pores. And it is semi-permeable and also called selectively permeable or differentially permeable, meaning that it allows only certain substance to pass through while preventing the other. That's we call it as selectively permeable. Whatever the cell wants, only it will absorb those. The remaining thing will not be absorbed or will not be taken. In the plant cell, an additional outermost non-living layer, which we call cell wall. Cell wall is mainly made up of cellulose. So the cell membrane in plant cell is surrounded by cell wall. So Robert Hooke is the one first observed only cell walls. In the dead cells of the plant, we have only cell walls. And it is mostly made up of cellulose. Cellulose is elastic in nature. And it gives the shape and rigidity. The shape of plant cell is mainly because of the cell wall. And it is freely permeable. If you dip in the oil will enter. If you dip in the water will enter. So usually the cotton is none other than the cellulose. So it will absorb both because it is a non-living thing, allowing the substance and freely permeable. It can enter and leave the cell without hindrance. Coming to the next part, it is cytoplasm. It is a part of cell inside the cell membrane and outside the nucleus. It is semi-liquid because many chemical reactions catalyzed by enzymes occur in here. It contains several organelles, each concerned with some specific functions. So it is semi-liquid in nature. Many chemical reactions. When the cytoplasm is chemically positive, then the meaning is the cell is living. It contains several cell organelles, each concerned with some specific functions. The part of cytoplasm other than the cell organelles, we call it as cytosol, which is mainly the liquid form. Only the liquid form of cytoplasm we call it as cytosol. Cytosol plus cell organelles is equal to cytoplasm, right? Cytoplasm plus nucleus is equal to protoplasm. Coming to the nucleus, it is a large, somewhat spherical body lying in the center of cytoplasm, right? Next, it is surrounded by double-layered nuclear membrane with nuclear pores actually. We have the nuclear membrane, nuclear pore, nucleolus, nucleoplasm, and chromatin network, all these things. And the space between outer nuclear membrane and inner nuclear membrane, we call it as perinuclear space. And next, the ground substance in the nucleus is the semi-solid, that is also semi-liquid form. We call it as nucleoplasm, just like cytoplasm. It contains one or more round-shaped stained particles, which is nucleoli. Nucleolus is singular, nucleoli is plural. Nucleoplasm contains a network of dark-colored fibers called chromatin fibers. So these chromatin condense to form some thick chromosomes during cell division. Only during cell division we have to call the things as chromosomes, right? And next, centrioles. So the centrioles will be moving apart during cell division. And see one thing, you can see the microtubules are coming from the centrioles. These microtubules are made up of tubulin protein that only will pull the chromosomes apart. We have the nucleus, cytoplasm, and these are the chromosomes. The meaning of chromosomes is colored bodies. The word chromosome you have to use only during cell division. Remaining time you can use the word chromatin material, means colored material. Coming to the next point, finer structures of cell, the organelles. The organelles are specialized membrane bounded structures in the cell concerned with some different functions. For example, ER will have other function, Golgi body will have one function, ribosomes will have one function, likewise. So we are going to see organ and organelle. Organ, just as these organs in the body, those are called organelles inside the cell. Those are very smaller than organs. That's why they named it as organelle, right? So the living parts of the cell which consist the nucleus, cytoplasm and other living bodies. Even the nucleus is also living. Collectively called protoplasm or protoplast. So the later term is protoplast, including nucleus, cytoplasm, nucleus, cell membrane. Altogether we call it as protoplast. Without cell membrane we call it as protoplasm. Okay. So appropriately use it for the plant cell for the removal of cell wall, that is protoplast. The plant cell without cell wall you have to call it as protoplast. The protoplast without cell membrane you have to call it as protoplasm. Okay. So this, for example, I'll tell you one simple thing. Plant cell, you remove cell wall, it will become protoplast. You remove cell membrane, it will become protoplasm. You remove nucleus, it will become cytoplasm. You remove all the cell organelles, then it will become cytosol. So the living plant cell, after removal of cell wall, after the living body is called organelles, are same in both plant as well as animal cells, except the cell wall in plants and centrioles and centrosomes in animals. So because cell wall is present for the plant kingdom mainly, and centrosome and centrioles in animals, they don't find in plants. The various organelles, nucleus, nucleolus, mitochondria, endoplasmic reticulum, ribosomes, Golgi bodies, lysosomes, chloroplast, vacuole, cell membrane, all are same. Coming to the main characteristic features and chief functions are cell membrane. We also call it as plasma membrane. In case of muscle, we call it as sarcolemma. In case of nerve, we call it as neurolemma. Main characteristic features. Outermost in animal cell, lies next to cell wall in plant cell. Very thin, flexible living membrane. Possess fine pores. In cell membrane, we have fine pores. It is semi-permeable and made up of lipoproteins, right? And separates the cell, contents of cell from the surroundings. It acts as a border, and it is selectively permeable or semi-permeable. It will regulate certain solutes, ions for entry of the cell. And in animal cell, the cell membrane will maintain the shape, but in plant cell, the cell wall will maintain the shape. Coming to the cell wall, you can find only in plant cells, because this chapter is dealing with plants and animals, so that's why we are talking only two. But even bacteria also have cell wall. We are not going in that way. We are only dealing with plant cell and animal cell. Non-living rigid layer surrounding the plasma membrane, mainly composed of cellulose, and it is freely permeable because it is dead material. Gives rigidity and shape of the plant cell. Allows the substance to enter or leave the cells without hindrance, means without any obstruction. Provides protection. Okay, cell to cell interaction. Right. Next, coming to the cytoplasm. All parts together inside the membrane, including the nucleus, contains the mixture of water, soluble inorganic and organic compounds and various organelles. Altogether is present in the cytoplasm. Different organelles contain, and it performs different functions. All the metabolic functions. Metabolism is the sum of all chemical reactions that is happening in the body. We call it as metabolism. Medium for early steps of respiration. Production of pyruvic acid. So anaerobic respiration, and the first step of aerobic respiration will takes place in cytoplasm only. Endoplasmic reticulum. So inner network of tubules, tubular double membrane. It continues with plasma membrane on the outside and nuclear membrane on the inside. It may be smooth or rough. If ribosomes present, it will be smooth. If ribosomes are absent, then it is rough. If ribosomes are absent, it is smooth. If ribosomes are present, then it is rough. Supportive framework for the cells, and synthesis and transport of proteins and fats. Mitochondria. Mitochondria is a semi-autonomous cell organelle, or which is double membrane bounded cell organelle. So various shapes like sausage, reticulate, oval, spherical, many shapes, and double walled. Inner membrane we call it as cristae. Have their own DNA consisting of several genes, and also contain their own ribosomes. Release of energy from pyruvic acid produced in the cytoplasm in the form of ATP. Seat of cellular aerobic respiration and stores energy. Synthesis of respiratory enzymes as well. Next, Golgi apparatus in animal cell we call dictyosomes in plant cell. Stacked flattened sacs. They contain tubules, vesicles and vacuoles. Tubules are flat, vesicles are round and vacuoles. The synthesis and secretion of enzymes, hormones, formation of acrosome of sperm, right? Next, coming to ribosomes. Ribosomes we also call it as Pellad granules because it is discovered by George Pellad, and that we call it as factories of protein synthesis, protein synthesizing factories. So smaller granules which is scattered in the cytoplasm, and it is attached to the endoplasmic reticulum as well. That makes the endoplasmic reticulum smooth, and coming to the single walled dense spherical bodies composed mainly of RNA. So they don't have any wall, it is mistakenly given here. They don't have any cell wall, cell membrane, and that is mainly meant for protein synthesis. Lysosomes. Lysosomes are the membranous sacs bud off from Golgi body. So we contain 40 different types of enzymes in lysosomes. It is mainly meant for intracellular digestion, means the digestion which has inside the cell, destroy the foreign substance. When cell is old or injured, it leads to autolysis and suiciding cells. So hence we call it as suicidal bags. Digest cartilage during formation of bones. Centrosomes, only found in animal cell. It is a region surrounding the centrioles located near the nucleus. We have 1 or 2 centrioles actually. And the centrioles are surrounded by microtubules. Initiate and regulate the cell division actually. Forms the spindle fiber with the help of asters. Coming to the plastids. Plant cell only. Several kind and most common ones are chloroplasts containing green pigments, chlorophyll. Double membraned proteinaceous matrix contain DNA. Disc-like structure called thylakoid contain chlorophyll. Chloroplast. Green they trap solar energy for photosynthesis, contains the pigment chlorophyll. Leucoplast, they store starch. Chromoplast, so they impart color to flowers and fruits, containing the pigment xanthophylls. And next, coming to the leucoplast, which stores starch. Chromoplast, color to the flowers, fruits, and they contain xanthophylls, which gives yellow color. Carotene, orange red color. Anthocyanin, which is dissolved in plasma, blue violet color. And next, coming to nucleus. So nucleus is the largest cell organelle and mostly spherical and dense. Nuclear membrane with pores to allow substance to enter and leave. Contains a network of thread-like structures called chromatin fibers which contain DNA. So regulate the cell functions. If removed, the cells will die. Contains chromosomes, bearers of genes that control the hereditary characters. So now the next thing is nucleus, which is the largest cell organelle, mostly spherical, dense, nuclear membrane with pores, so that allow the substance to enter and leave. Contains network of thread-like structures called chromatin fibers, which contain DNA. So regulates the cell functions. If removes, the cell will die. Yes, if nucleus is removed, the cell will die. But there are some cells without nucleus. So certain chromosomes, bearers of genes that control hereditary. For example, RBC, the lifespan of RBC is very less because there is no nucleus and there is no mitochondria also. If you go with sieve tube cells, they don't have nucleus. Actually, it is controlled by the nucleus of companion cells. Next, nucleolus. So that is one or more round nucleoli inside the nucleolus. The nucleolus is the part where the protein synthesis is formed by storing the RNAs. So they produces ribosomes and dictates the ribosomes to synthesis proteins. Okay, when there is more nucleolus, that means more ribosomes. More ribosomes means more protein synthesis. Chromatin fibers. The network of resting stage of the nucleus condenses the chromosomes during the cell division, made up of DNA threads. Okay, so chromosomes carry hereditary information or the genes. So next is vacuoles. Vacuoles are the clear spaces with water and other substance in the solution. Plant cells have larger vacuoles, while the animal cells have fewer and smaller ones. So plant cells usually have larger vacuoles actually. The vacuole membrane we call it as tonoplast, and the fluid we call it as cell sap. So they store water and other substance, food, pigments and base products. So they give turgidity to the plant cell by pressing against the cell wall. And finally granules. Granules are small particles, crystals and droplets. Starch in plant cell, glycogen in animal cell, and fat containing granules serves as food for cell. Next topic వచ్చి ప్లాంట్ సెల్కి యానిమల్ సెల్కి డిఫరెన్స్. ప్లాంట్ సెల్కి సెల్ వాల్ ఉంటుంది, మేడప్ ఆఫ్ సెల్యులోజ్. యానిమల్ సెల్కి సెల్ వాల్ ఉండదు. సెంట్రోజోమ్స్ ఆర్ absent in plant cell, centrosomes are present in animal cell. So vacuoles are prominent, one or more. Animal cells lo vacuoles is small, temporary, concerned with excretion or secretion. And coming to the plastids, usually plastids are present in the plant cell. Animal cell lo plastids undavu. Size, plant cell lo usual ga peddadi untundi, animal cell lo size usual ga smaller ga untadi annamata. Plant cells peddavi, animal cells chinnavi. Cytoplasm is not so dense in plant cell ఎందుకంటే మేజర్ పార్ట్ లార్జ్ సెంట్రల్ వాక్యూలే ఆక్యుపై చేస్తుంది. యానిమల్ సెల్లో అలాంటి లార్జ్ సెంట్రల్ వాక్యూల్ ఏమీ ఉండదు కాబట్టి cytoplasm will be denser and more granular. Arrangement of cytoplasm, only a thin lining of cytoplasm mostly pushed to the periphery. ఇక్కడ cytoplasm వచ్చి ఎంటైర్ సెల్ మొత్తం ఉంటుంది. ఎపిడర్మల్ ఎలా ఎగ్జామిన్ చేస్తారు? హౌ ద microscopic examination of onion peel. Onion peel is for plant cell. Epidermal cells of onion. So epidermal peel from onion is easy to prepare. So we have to take the outer covering of onion. So the peel of onion. So you can proceed. Cut the onion bulb into 4 quarters lengthwise. Afterwards, from one of these quarters, remove one thick scale leaf. Take the fleshy scale of the onion into your hand and tear it from inside, means from the concave side, so that you will get a thin transparent strip. So you can try it also. Usually you will get thin transparent strip. Using a pair of forceps, remove that strip and put it in the watch glass containing water. So now cut the piece of the peel about 5 into 5 mm and mount it on the slide in the drop of water, so as shown in the figure. So cover the peel with a cover slip carefully so that tissue does not get wrinkled. Examine the preparation under low power microscope. So you may as well stain the material with iodine or eosin solution, so which will make the nucleus more distinct. So ఈ పిక్చర్లో చూసినట్టు ఆనియన్ని 4 క్వార్టర్స్గా కట్ చేసి ఒక సన్న స్లైస్ తీసుకొని, ఒక సన్న పీల్ తీసుకొని దాన్ని కాన్కేవ్ సైడ్ కట్ చేయాలి. కట్ చేసినప్పుడు మనకు ఒక పీల్ వస్తుంది. అది 5 into 5 mm డయామీటర్లో కట్ చేసుకుంటే మనం దాన్ని ఈ గ్లాస్ ఈ స్లైడ్ మీద వాటర్ వేసి దాంట్లో ఈ పీల్ పెట్టి దాని మీద మనం స్లిప్ పెట్టాలి. సో లేదా స్టెయిన్ కూడా చేయొచ్చు. సో మోర్ డీటెయిల్స్ ఆఫ్ సెల్ స్ట్రక్చర్ విల్ బీ సీన్ అండర్ హై పవర్ మైక్రోస్కోప్. Cells of onion peel have somewhat regular shape, linear or rectangular. Each cell has a prominent cell wall, a nucleus, and a cytoplasm. Encircling the one are 2 large vacuoles. The details are better in high power microscope, especially the thick cell wall. So note that the cells are firmly bounded together. Nucleus is placed towards one side, which is usual case of most of the plants because of the large central vacuole. So nucleus is the key of life. Yes, although every part of the cell contribute to the life of the cell as the whole, yet the main performer or the master of the cell is nucleus. So without nucleus there is no further growth of the cell or further reproduction to cell division. The cell will be having very shorter lifespan as well. So this can be easily experiment by experiment performing on amoeba, which is single celled organisms. Nucleus is the main thing for division. Without nucleus, there is no division at all. You can see this amoeba, which is single-celled organism, comes under the kingdom Protista. So it will divide into 2. 2 will divide into 4. 4 will divide into 8. This we call it as fission. If the nucleus is removed from amoeba, then the amoeba will die. So you can see here. So if the nucleus from another amoeba is transplanted in the enucleated amoeba, the recipient will survive and the dies while the donor amoeba, whatever the donor amoeba will die. Whatever the amoeba will receiving the nucleus will live. So you can see an experiment to show that nucleus is essential for normal life. So you can see here some of the cell organelles of an animal cell. So you have to identify what it is. And the first picture is centrioles. And the second picture is mitochondria. And we have the Golgi bodies, chromosomes, and we have the endoplasmic reticulum here. And here we have granules. And here we have the last one is chromatin fibers. Right. So this is all about the chapter of Icse class 10 cell, the structural and functional unit of life. Yeah, thank you very much. Bye bye. Please don't forget to subscribe to my channel for more biology videos.